I think the role of the learned society in science education and development is one which everyone should appreciate. I explained earlier really that when I went to the University of Bath, not many people were interested in discussing research, and it was really during this time that I became seriously interested in learned societies, because they seemed to me to be the last place you could go where you could actually talk about science with other practitioners in an open and constructive way. I'd always been an attendee at meetings of the PhysSoc and also the PharmSoc, but during my time at Bath, I began to take a very serious interest in the Pharmacological Society. In those days, this was a much smaller society than it is today, but we used to have 4 meetings a year, and in those days, pharmacologists used to go to all 4 during the year. One was encouraged to do that as part of one's training. So the result was you knew most of the people who went to the meetings on regular occasions. You may not remember their names, but you could certainly recognise their faces. You knew where they worked, you knew roughly what they worked on. And the other thing was you always tried to put in an abstract at each meeting, and this was another very important aspect of your training, as was of course presenting an oral communication in exactly 10 minutes and fielding questions from some of the top brains in the field. But it was more than that, because these societies provided a sort of a cradle for the development of younger members, and more senior members of the society often acted as mentors for younger members, and would often, for example, telephone them and say, have you seen there's a good job going that would just suit you, or would you like to come to my lab? I've got a demonstration of another bit of equipment. It was very, very friendly and open, and there was a lot of interaction between those two halves of the membership. I'm afraid a lot of that seems to have disappeared now, and there are various reasons for this. Many learned societies have reduced the number of annual meetings, so that's one reason, so you don't get to see people so frequently. The second reason, I think, is that there are so many more meetings to go to these days. For example, when I started out as a young pharmacologist, you went to 4 meetings of the BPS per year, and to IUPHAR once every 3 or 4 years, and that was your quota of meetings. But now there are so many, no one can afford to go to all of them, so you have to be very selective. And I think the important thing about learned society meetings, and probably about all meetings actually, these days anyways, you don't go there necessarily to learn things, because if that's the case, you might as well stay at home in your office and read papers on your PC, or download something off PubMed or something and read it. The reason you go is this exchange of information, ideas, students, techniques, transgenic strains of animals, and it's the human contact. Science at the end of the day is a human activity. Especially these days, I think the days of a lone scientist have probably gone. Maybe not completely true, but certainly largely true. And most science is done in groups these days, and actually often in worldwide consortia. Just look at some of the genetics papers that have come out recently with 100 authors or something. So I think the nature of science has changed. I think the function of meetings has probably changed. And I'm just sad in a way that perhaps so much of the original culture has disappeared or is disappearing. When I became more involved with the BPS, I agreed to take over the role of an officer position. So in those days, the society was organised in a completely different way to the way it is today. It didn't have a permanent headquarters. The officers were distributed around the country. There were no permanent staff, so everything had to be done using the help of your students, your postgrads, your postdocs or postgrads, or else your PA if you have one. So the first job I took over, the first major job I took over, was meeting secretary, and this was a 3-year appointment, which involved organising 4 meetings a year for 3 years, and I had to travel around the country, seeing people, checking up on their arrangements for their meeting. I saw all the abstracts that were produced. I helped sort them into piles. I helped construct the programme, and I vetted the entertainment suggestions and so on. It's quite a demanding job. And in those days, once you were meeting secretary, you automatically went on to serve a further 3 years as general secretary, which was the old name for president, as it's called now. So this was a further job with different responsibilities, including the general strategic direction of the society, and often chairing many meetings, and most crucially of all, reading the social minutes after dinner at every meeting of the society, which again I'm afraid is a nice tradition which has unfortunately gone by the wayside. But these years were, although they were very, very demanding, but in those days it was considered part of your overall obligations as an academic to help run the scientific infrastructure. In fact, everyone was expected to play a role in running the national science infrastructure. And so it was not exceptional to do these jobs. Nowadays it seems to have changed, and everybody seems to be so much more accountable for their time that any outside commitment that doesn't immediately result in grant money flowing in is rather frowned upon in some circles. But it never used to be, and I think the society was all run by volunteers in those days, and it functioned perfectly well.
The BPS is something that, in a sense, I've grown up with, because it was considered to be absolutely a part of being a pharmacologist in all the departments I've ever been in. And so it was, not a matter of if, but when would you become a member? When would you give your first, in those days you had to give at least one communication to become a member, and when would you do that? And in the department at the College of Surgeons, it was a big event, because everybody who was up to give a paper would be drilled, and they would have to give a rehearsal, and people would ask the most unpleasant questions, purposely, saying, okay, you've got to be ready for something way out from nowhere that you've never thought about before, and you've got to be able to handle it somehow. But what you mustn't do is to pretend it doesn't matter. If somebody asks you a question, you have to treat them seriously, even though you have to say, that was a nice question, but I wish I knew the answer. I just have no idea. You can't say, oh, it doesn't matter, because it's rude and it's not scientific. The rehearsals were absolutely important, intrinsic part of the whole business. But training in pharmacology was not just doing the experiment, it was communication. You had to get to communicate, you had to tell people about your results in an understandable way, and then handle the questions in a sensible way. Too often I felt other people would react almost with hostility to a question. You asked them a question and they seemed to believe it had some ulterior motive, when certainly for me very often the matter of sheer ignorance of the subject. In the department of the College of Surgeons, we had this very strong tradition of having foreign postdocs, and Sergio Ferreira, Salvador Mancada, Arnold Herman, and Americans also, of course, but none of them were familiar with the particular experience of giving a paper to the PharmSoc. And of course, for those whose native tongue was not English, it was a double problem. And So we used to rehearse not just what you said, but how you said it and all that sort of stuff. And it was a very serious, undertaking, and a lot of time and effort and preparation went into producing a communication for the BPS. And later on, of course, when you were a little bit better at it, you would start asking questions about other people, you know, about how they were doing their work, what's happening, what would happen if have you tried this, have you tried the other, and so it was. I remember Sergio Ferreira was very much involved in going to meetings, giving papers, participating in the questions, and of course in the bar afterwards, where so much good pharmacology is done and planned and discussed. This was the place that, I mean, BPS meetings were immensely valuable in talking about problems, getting ideas from people, and everybody would put their tuppence worth in. And you did it. It was an important part of your science. It was an important part of your science. It wasn't an add-on. It was an important part of the science that you were doing.
I think as we move through life, there are obviously key people who are important to us in our development, who nurture us, who take us under their wing, who basically give their knowledge freely with regard to what we're trying to achieve as our personal goal in life. Certainly, the British Pharmacological Society has played a tremendous role for me in meeting many famous pharmacologists, scientists who have basically given so much of their lives to developing new drugs. Sir James Black being one of them, who was a tremendous influence on my life and how the development of beta blockers, which my wife is actually taking now. So people like Sir James Black, people, the management at ICI, people like Barry Cox, Mick Turnbull, then I move into the British Pharmacological Society, Norman Bowery, Rod Flower, Ray Hill, the staff, so many good people with regard to their passion for the science, their passion for helping young people, bringing young people along in the science. And I think that's the thing that I've always tried to bring back to my life. As a young pharmacologist, I found the BPS meetings so important in my career and my life, in both understanding the science, understanding drug development, understanding how scientists think. Many eminent scientists of the day used to sit and ask questions. Now, as I've moved on in my career, I'm asking those questions with regard to young scientists. And to see young scientists at pharmacology meetings and to see their enthusiasm and adopting many of these new techniques to advance neuropharmacology or other aspects of pharmacology is just amazing. And I think it's a great opportunity, as it always has been, for young scientists within the British Pharmacological Society or other learned societies to move forward with their careers and gain the knowledge that people like myself had gained over many years. One of the things I would say to a young scientist coming through with whatever learned society is always challenge the dogma. Because here you are standing up in front of your peer group, basically explaining your experiment. You know that experiment better than anyone. You know the data better than anyone. So therefore, I think it's extremely important, and never be put off by the audience or people around you. Explain it, present the data, and answer the questions. One of my first presentations was in Dublin many years ago, when the chairman had fallen asleep because he'd been at the official dinner the night before and perhaps had drunken a little bit too much wine. He'd fallen asleep in my presentation. I'd finished my presentation and I had to turn to him and wake him up. I was looking around the audience, which was very few, because again it was a Friday afternoon, one of the last speakers of the day, and there was only one person in the audience who put their hand up to ask a question, and that was a professor from Edinburgh. And he asked the question, and I was so pleased, I was so excited, and I gave him an answer which he accepted. So that was my first presentation to the Pharmacological Society, one that I will always remember and always cherish. And this is what you have to look forward to in the future. Hopefully you'll have a few more questions and a chairman who doesn't fall asleep.
So eventually he would get me to prepare a talk, 10-minute talk for the British Pharmacological Society, and I would prepare the talk. I would start off and I'd get about 3 words through, and he'd say, no, what do you want to try and say? We'd go through that. So what do you want to say is this. So that's your first line, learn your first line, get accustomed to where you are. So right, you've learnt your first line. I'd start my first line, I'd probably get through about 3/4 of the way. No, stop that. You should have done that. Go back and do it again. I'd get through the first line, I'd be on the second sentence. No, you've got that wrong. Go back, do it again. Third line, go back, do it again. Go. Stop waving your arms about, you're distracting me. Hold your arms like this. Go back and do it again. No, use your pointer properly. Point on, off, like that. Go back, do it again. But to cut it short, and it wasn't short, we would be rehearsing that 10-minute talk for the vast proportion of that day, by which time I knew it worked perfectly. I knew where to wave my arms and when not to wave them. I knew when to use the pointer. I'd learned my first sentence. I'd learned to say, Mr. Chairman, ladies and gentlemen. I'd learned to say thank you. I'd learned everything. And it took most of the day on the first one. The subsequent ones we did were less, but we still had to rehearse it. And I still do the same to my students.
I've just come from the latest British Pharmacological Society meeting, where we've seen our membership approach 4,000. We have a lot of young people. It suggests to me that pharmacology is going to have a big place in UK science for a long time to come, partly because I think now we are trying to make sure we interact with other societies as much as possible. We're holding more joint meetings with the British Association for Psychopharmacology, that I've also been much involved with the Physiological Society, with the American Society. So I think going back to John Gadham's comment that the pharmacologist is a jack of all trade and a master of none, you know, we're following his ideas from 40, 50 years ago and providing a pharmacological approach to everyone's science, and that has to be good. British Pharmacological Society has always been a major part of my life. I joined in 1973 when it was very much a career step, that after you got your PhD and you'd published a little, you would be expected to join the society, and it was the main to use again a current technique, networking opportunity you had. Could also be quite frightening because you had to present orally. There weren't such things as posters, and people would ask you questions, and since the abstracts were going to be published by the journal, would correct your abstract and make sure that it was correct even down to the last comma sometimes. And I was fortunate, I got involved in helping to organise the 1981 meeting, the 50th anniversary meeting, and I think that led on to me being offered the position as meeting secretary, and that in turn you were expected to move on to becoming the general secretary, which is now called president. I rejoice in the figure, in the title of President Emeritus. They made it up to us by only being called a secretary. Something I enjoyed enormously because I met huge numbers of people outside my own area of interest, and got to know a large number of pharmacologists in the country. And recently went back as a member of council and a trustee because I still enjoy the society and felt it would be fun to be involved at least a little in decision making. But the society has changed enormously. It was a society primarily for giving abstracts and oral and poster presentations 4 times a year, which was a number of meetings I used to have to organise, because it took a long time to publish a paper in the 1980s, and now from acceptance to submission to acceptance to publication you can get something out in a couple of months. In those days it would sometimes take a year, so people wanted to establish primacy of discovery by presenting to the BPS. My very first oral presentation was actually to the Biochemical Society, who met at the Institute of Psychiatry down in Denmark Hill, and at the end I was asked a question about the levels of serotonin metabolites in the CSF. I of course was working just on rats, and said that no, I hadn't done anything, but there had been some really interesting work going on by Ashcroft and Eccleston in Edinburgh studying that very thing. And after I had finished, the person asking the question saying thank you very much, very politely, I went off to see my supervisor, who said, oh, that was a nice presentation, and you know who asked that question? No, I answered. He said, that was George Ashcroft, and I was fairly mortified, but Joe pointed out that all scientists like their own work being quoted back at them. In 1987, Paul Van Hoot came to England to help set up the Serotonin Club. He decided that rather like the Catecholamine Club and the Histamine Club, there should be a club of people interested in serotonin, which is really rather late when one thinks about it. And we met, I think, in the lecture theatre. Anyone was interested of the School of Pharmacy, and he gave a talk about what he wanted to do and how he wanted to set it up. And then I have to say, as far as I was concerned, totally out of the blue, he said, well, Richard Grigg could be the one of the British officers. So yes, thank you. And I then moved on to being vice president and president. Wasn't sure because I was right in the middle of my activities with Astra in those days, so I'm not sure that I was as active as I ought to have been as president. So when the job as treasurer secretary came up, I said yes, I'd do that, and I did that then for about 10 or 12 years, I think, which was very enjoyable, because I had to write a newsletter and help him get involved in organising meetings. We always ran a meeting at Christmas time as a sort of offshoot of the British Pharmacological Society meeting, because a lot of people, from Pat Humphrey to Danny Hoyer, there was a very strong serotonin research in the UK and Europe. John Fozzard is another that should be mentioned, and so we always had a lecture and a dinner on one of the quieter evenings of the BPS meetings. Enormous fun. Recently, I asked actually when I was secretary whether we ought to change the name, because Serotonin Club sounded a bit informal when we were trying to get money to help support the meetings, and everyone, including Paul Van Hoot, said no, they liked the name. A few years ago, about 3 or 4 years ago, almost led by Paul, I might say, a move was made to change the name, and it's now officially called the International Society for Serotonin Research, ISSR.
So I think one of the seminal activities in starting up the 5-HT classification studies worldwide was definitely the Heron Island meeting in 1987, I would say. The Serotonin Club had been started by Paul Van Hoot in 1986, and him and I talked a lot about classification, and he was very keen on it and wanted to support it. In fact, Paul found a lot of the money to subsidise the Heron Island meeting, but equally Glaxo Australia put quite a lot in, because they knew obviously that we were about to launch sumatriptan in the not too distant future, and so that was the sort of means of the funding. Ewan Milacrane was the prime mover in organising the meeting and asked me to help, and I said, well, I don't mind as long as it's somewhere decent. And he said, is Heron Island decent? I said, right, you're on, I'm helping you. So off we went, and it was a very exciting meeting. We had a tremendous opportunity to go snorkeling and fishing and all those sorts of things. But I can tell you that during the meeting periods, everybody was in there, because it was just one of those meetings where it was buzzing and everybody wanted to know how many 5-HT receptors there were, and whether the name they'd given to their pet 5-HT receptor was the right one. And I mean, it was just an amazing meeting, and it was well organised, and I think the climax in terms of classification was the round table discussion where everybody in the conference was involved, and I and Brian Richardson chaired it, and everybody had their 6 penny worth, and in the end Brian and I wrote something up together. And the whole point of that was that I wanted everybody to be involved, everybody to feel part of a scientific fraternity worldwide, where they could have their views expressed. And some people were not totally happy with the outcome. I know Michael Gershon, who's a good friend of mine, I love him dearly, but he was very upset these 5-HT1P receptor was not allowed, and it still isn't allowed. So maybe we got it right, I don't know, but we'll have to see. But it was rather funny that evening. Everybody was still very high and got in the bar, and people were having a drink, and then somebody decided they wanted to take a photograph of some of the key players, and everybody had to put their hands up with how many receptors they thought there would eventually be. And so we all had to, there was, right, now you go. And I put my hand up, and I only had one finger up. Some people had 10 up. And what I really meant was one finger meant we found the receptor that's going to provide a drug, and it's sumatriptan. So that was funny. Another thing that wasn't quite so funny was when we were leaving. We had 3 big boats, massive big launches, to take us back to catch our planes. We had to cross the Coral Sea, and the one I was in with my wife and other people broke down. There we were in the middle of the Coral Sea. I can't really claim there were white sharks circling around, but it was quite eerie being in this in this boat that was not going to go anywhere, so they had to get lots and lots of small boats to get us back to the island, and then we had to have helicopters to take us to the airport before we missed our planes. So that was all good fun, but a lot of people remember that as well. But it was quite a meeting. I think it was very successful scientifically, and it was great fun, and something I don't think anybody who was there will ever forget.
Another important sort of feature of my career has been my involvement with various organisations. I've been involved with the British Pharmacological Society, the International Club for Serotonin Research, I think it's called today. And also I was involved in the establishment in the monitoring of molecules in neuroscience grouping, which is now an international group. But probably my most important involvement was with the BAP, the British Association for Psychopharmacology, of which I had almost all, I think, the council jobs that were in existence at my time, and I ended up as the president. And after coming off council, I was involved with the governance panel as well. I was the chairman of that for a couple, 3 years, the role of which is to make sure that the association is responding to the needs of its members as well as observing the laws of the association. I think such groupings of scientists are very important because they do allow crosstalk between different types of individuals. So with the British Association of Psychopharmacology, there are both clinical psychiatrists and basic scientists, and their meetings give a great opportunity for a good exchange of views and ideas which can improve the translational value of the research done by both sides. And I think that's one of the great roles of any organisation, I think, to allow good crosstalk, and that can lead to new research ideas, new research projects.